Maria here will record. Got it. Yeah, perfect. So Caio is just a quick introduction, is a postdoctoral research fellow at the Indiana University, now with Olaf Sporns. But then before that, he obtained his PhD in systems neuroscience from the University of Melbourne, and then stayed there for a postdoc, and then followed another postdoc in Sydney. And then his focus is actually mostly on these brain network communication models, and that's good because now I think, I'm just guessing, but I think he will just give us one crash course onto the whole thing and what are the concepts and which models are there, and then what sort of applications do we have. I hope, I mean, I'm really reading your title, but I hope I got it right. Not empty promises, at least. But yeah, I mean, let's dig in.
Thanks, Kason. It's a pleasure to be here and speak to all of you. So yes, my research is on network neuroscience, and more specifically on brain network communication models. And today, as Kason said, is going to be a little bit of a crash course and introduction to brain network communication models. So if you have any questions at any time, please feel free to interrupt. No problem at all. So I like to motivate the use of these types of models by arguing that they provide a bridge between brain structure and function. And I'm going to show you two examples of what I mean by that. So the first one is this figure by Laura Suarez and colleagues in a recent review. And here on the left, we have a macro-scale human connectome. Oops, apologies. Macro-scale human connectome. So here, the nodes in this visualization are gray matter regions, and the links between nodes are white matter tracts. And we can express this in the form of a structural connectivity matrix. So this is mapped with diffusion MRI and white matter tractography. So these are anatomical projections, right, between regions. And here on the right, we have a functional connectivity matrix, which is a measure of statistical dependencies between the activities of regions, right? So this is measured from correlations from fMRI bold signals. And the point that they're making here, and they're certainly not the first ones to make this point, but they illustrate it very nicely, is that most regions, most gray matter regions, are not connected by an underlying physical connection. And yet, they can show strong functional connectivity, right? So only a small fraction of regions are anatomically connected, while lots of regions, many more than those, show strong functional connectivity. Another example in a similar vein comes this from beautiful recent paper in Science, which is the paper that mapped the connectome of the fly larva. So here's kind of what the neurons look like. This is more than 3,000 neurons. And similarly, you can arrange the connectivity between these cells in an adjacency matrix. And this is really cool because it lets us study sensory motor pathways from what they call input regions. So these are regions that receive input from the environment to output regions. So these are effector neurons that might be related to controlling muscles in the fly larva. And what, again, is really cool is that these neurons are not directly connected by a synapse, right? This sensory motor pathway depends on a polysynaptic cascade of signals that happens on top of this anatomical connectivity. So the point that I'm trying to make here is that brain function depends on polysynaptic communication that unfolds on top of a structural connectivity. And the idea of brain network communication models is to use the tools from network science to understand how these neurocommunication processes are shaped and constrained by the underlying anatomical connectivity. So what do I mean by network communication models? There's many computational approaches in this space. Sort of the space of neurocommunication is very broad. And different people have tackled this problem from different angles. You can look at it from a dynamic systems perspective. You can look at it from an information theory perspective. You can look at it from a network controllability perspective. For this talk, I will consider very simple network models that provide an alternative to these other approaches mostly based on graph theory and network science, right? So the trade-off that these models make, because they're so simple, they typically abstract away neurobiological detail. But what they gain in doing that is they become very general. So they give us a general mathematical framework to understand a lot of seemingly different neurocommunication processes. Because of this, they have more of a phenomenological flavor than a mechanistic flavor. And I'm going to talk after I show you some examples. I'm going to get more into these details. So the kind of structure of the talk is going to follow this recent paper that we published. And this is a review on all of the topics that I'm going to approach here. So if you're interested, read the review for more details. Okay, so let's start with some really basic concepts. Here I have a structural connectome again. The nodes are white matter regions. The connections are, the nodes are gray matter regions. The connections are white matter links. And a walk in this network is any sequence of nodes and connections between two nodes. So here we have nodes I and J, and this here is a walk between them. And a walk doesn't impose any constraints on revisiting nodes and connections. So here you can see that this walk revisited this node, right? So it goes from I to J. A path is a walk in which we don't revisit any nodes or any connections, right? So this is a path. And the shortest path between I and J is, quote unquote, the most efficient path between I and J. So it's the one that lets you travel from I to J with the smallest cost. And we're going to go into the detail of what I mean. But intuitively, we can think of this kind of communication channel length, and we can see that some channels are quite short or efficient, and other channels are long or inefficient. And kind of intuitively, we think that short communication channels would be better to integrate information between regions as opposed to long communication channels. Okay, so what are the basic ideas that people have thought about in this space? So if we want to go from a source node S to a destination node D, kind of one very broad idea that we can have is this family of routing protocols. So the idea is that a signal is going to be passed from the source to a next node, then to a third node, then finally it reaches the destination. So these routing protocols, they kind of assume that communication unfolds along a single path through the network. Another flavor of communication is this idea of diffusion processes. So instead of following a single selectively accessed path, communication is more of a diffusion process that unfolds along multiple network fronts. So here from the source, you would go to these two nodes. From these two nodes, you might go to this one, and then eventually you reach the destination, right? So you have these two kind of very different conceptualizations and very broad conceptualizations of communication processes on networks. And here I'm going to argue that stemming from these two very broad families, we have a lot of different communication models, more specific models that have been proposed. So we have diffusion processes that have more of a stochastic flavor. They unfold along multiple fronts of communication. And on the other end, we have single paths unfold along selective pathways, and they have more of a deterministic flavor. So these families, and then I'm going to talk about these parametric models that are sort of in between. These families are broad conceptualizations of neurosignaling. A model takes this broad conceptualization and provides a concrete strategy of how to propagate a signal or identify a path in the network. By far, in network neuroscience, in applications of connectomics, the most common way to model communications is via the shortest path. So we have two nodes and we identify, let's say, the smallest number of hops or the smallest number of intermediate regions from which we can integrate information between these two nodes. And this measure, shortest paths, is really the basis of a lot of graph theory measures that we use in network neuroscience. So if you have thought about small world networks, characteristic path length, global efficiency, betweenness centrality, all of these measures have baked into them the idea that communication happens exclusively via the shortest paths between regions. And this kind of makes sense because from a sort of biological perspective, this would mean the most efficient way to transmit signals. And it would also be very metabolically cheap because you're minimizing the number of kind of synaptic junctions that you're crossing between regions. The problem with this model is that it requires global knowledge of the network. So what I mean by that is to identify the shortest paths between two regions or between two cells, you actually need to know the entire network, all connections between all nodes. Otherwise, you're not guaranteed to find the shortest path. So algorithmically, it's hard to see how the brain would have a mechanism to implement communication following this strategy. On the other end of the spectrum, so totally different, is the idea of a random walk. So a random walk essentially proposes that you would send the signal by randomly sending it to one of your connected neighbors, right? So you just randomly send the signal along, and eventually it will reach a certain destination. So the random walk suffers from the opposite strengths and weaknesses of shortest paths. So it would be very inefficient, it would be energetically expensive, but on the other hand, it's very plausible to implement this mechanism in a biological system. You don't need a lot of information about things that are far away or centralized information about the network. So it requires local knowledge. So these two conceptualizations of network communication form this kind of spectrum from decentralized ones, so that means you only need local knowledge about who you're connected to centralized ones where you need knowledge about the entire network to send information. And in between these strategies, a lot of different models have been proposed that, you know, they fall at different places of this spectrum. And we have these three dimensions of communication cost, as I call them. We have the delay, so how efficient information can be transmitted. We have this idea of information. Does it require only local knowledge, which is more plausible? Does it require global knowledge, which is less plausible? And we have this idea of energy. So what are the metabolic resources necessary to send signals according to a certain network strategy? And these three dimensions provide a way to sort of conceptually evaluate different models. And I'm going to be using that throughout the talk to explain a few different models. And finally, we have the models, and we also have network communication measures. So the difference between a model and a measure is that a model proposes a strategy to propagate the signals, whereas a measure is a way to quantify communication under a model. And therefore, each model, there's many different ways to quantify communication. So for instance, if you're familiar a little bit with graph theoretical measures, shortest paths, so the idea that communication happens via the shortest paths, can be quantified by the characteristic path length, but it can also be quantified by global efficiency, which are kind of the same thing. But this is what I mean here. And I'm going to explain this a little bit more in detail later. These measures come in a lot of different types. They can index efficiency, length, information, resilience. I'm not really going to get into the details, but there's more on that on the review. Okay, so now I'm going to give you some examples of network communication models that have been proposed to kind of build a little bit of intuition of how these things work. Okay, so the problem with shortest paths is that it requires this idea of centralized knowledge, that we need to know the entire network in order to route a signal efficiently between two regions. But what if we didn't need this, right? So this is some work that I did in my PhD on network navigation. So navigation proposes to propagate information based on local knowledge of a distance between nodes. So if anyone here did psychology undergrad, this is sort of inspired by the work of Stanley Milgram on sending letters between people in different states in the United States. And the rule there is that you had a recipient that you had to reach with your letter, but you could only send the letter to someone you knew on a first name basis. So this first name basis requirement defines the structure of the social network in which people were embedded. And what people did is they forward the letter to people that lived closer in distance to the target they wanted to reach, right? So this already gives you a bit of an intuition of how this is going to work. So similarly, the brain is a spatially embedded network. So here, the measure of node distance that I'm going to use is just the Euclidean distance between gray matter regions, right? So how far away they are in a straight line in 3D space. So the rule is very simple. If you have a signal, if you're the source, you progress to your neighbor, so a node that you are directly connected to, that is closest in distance to the target. So this is a very simple example in this toy network. Imagine this is embedded in a two-dimensional plane. If we want to go from A to H, right, this is our goal. If you follow the shortest path, you would go from B, then to E, then to H. This is three hops in the network, 3 connections. But if you want to navigate, what you would do is you want to send a signal to the neighbor that is closest in distance to where you want to go. So D here is actually closer to H than B. So you would send it to D, then from D to F, F to G, and then G to H. So here it took us 4 hops in the network to reach H, right? So we set out to see is this an efficient strategy to communicate information in brain networks? So to do this, we proposed this measure called the efficiency ratio. And all that it does is its benchmark is navigation versus the shortest path. So again, by definition, the shortest path is the quickest way you can travel the network. So in this example here, which is very simple, the shortest path took 3 hops, the navigation path took 4 hops. So this is 3 over 4, 75% of, let's say, optimal efficiency. And then we can do this for every node pair in the network, and we can make an average, right? And this is kind of what we found. So along these axes, we have the connection density of the network. We can kind of threshold that at different levels. And here we have the efficiency ratio. And what we found is that for network densities, kind of where we reasonably work between, say, 10% and 30%, this really simple strategy that doesn't know what's going on in the whole network, as opposed to shortest paths that knows all the weights of all the connections of how everything is connected, can actually identify very efficient paths. And if we either rewire the network or we reposition the nodes in space, we no longer can communicate information efficiently. So what was cool about this paper is that it proposed this 3-way relationship between brain network topology, how things are wired together, geometry, how things are positioned in space, and communication. And the key point here is that navigation, it has weaker knowledge assumptions than shortest paths, with almost the same efficiency, so the same sort of transmission delays. The problem, of course, is that justifying how a region knows how far away it is from a certain target is still really hard. There's some people doing this in plant network science, which is really cool. And apparently they think that this is a justifiable assumption. But I think in the brain is still a bit iffy. So the point here is not to say that this is how the brain is communicating information, but it's just to demonstrate that there could be different mechanisms or different rules in which you can achieve efficient communication without necessarily global knowledge of the network. So another model that is very popular is this idea of broadcasting. I call it broadcasting. Lots of people call it communicability. This is more of a terminology thing. So the idea here is that signals are transmitted via all possible walks between source and target nodes, which is very different, right? Now, we're leaving this idea that we follow very selectively accessed paths to more of a conceptualization of communication via diffusion, right? So what do I mean by that? All possible walks, right? So let's say we started this node here. Walks of length 1 just means the nodes you're directly connected to, so all your neighbors. So at step 1, you send the signal to all your neighbors. For walks of length 2, you send a signal to all nodes that are 2 steps away. But that also includes back to yourself, right? You send it to a guy that you're connected to, and then it sends back to yourself. Or it also includes this kind of path where nodes that have already received the signal in 1 step receive it again. So it's really all walks of length 2. So by walk, I mean that you can repeat which node has already received the signal. And for walks of length n, the number of walks between a source and a target is given by the exponential of the adjacency matrix. So what we want here is the intuition is that short walks between nodes would be sort of strong candidates for channels of communication, whereas very long walks wouldn't really carry a lot of weight. So we want to discount the contribution of long walks and prioritize the contribution of short walks. And the way that Estrada and Hatano that proposed this in 2008, they proposed this factor proportional to the factorial of the length of the walk to discount the contribution of long walks. And this kind of converges to this matrix exponential, which is very nice. So that means you can compute this analytically. So the nice thing about communicability is that this strategy is very simple and it doesn't mandate any knowledge of the network topology at all. You're just doing it in a very kind of dumb way. But because you're sending it along all possible walks, this set of all possible walks include the shortest paths between two nodes. So if you're thinking of transmission delays, it's actually very fast. It's as fast as shortest paths to go from one node to another. The problem here is that this strategy is very costly, right? From a metabolic point of view, if you're endlessly sending these signals back and forth, that would require a lot of energy. So this kind of illustrates how we can devise a strategy that is very efficient in terms of transmission delays. It doesn't require a lot of information. It could be reasonably implemented in a biological setting, but it's prohibitive because we require too much energy, sort of in a conceptual way. So this is the idea of communicability. I would say that it's, after shortest paths, is probably the second most used measure in network neuroscience. And the last measure I want to tell you about is this idea of biased random walks, which I think is really cool. This is more of a newer concept in network neuroscience and brain networks. I'm going to try to explain how it works to you. The idea is to bias random walks to make them more efficient. So usually a random walk just pings around a network in a random fashion. But what if we can influence this pinging around so that it's a little bit more efficient? So we can introduce a parameter that endows the random walk with the ability to choose edges that are more aligned with the shortest paths between two nodes. So let me try to explain how this works. So imagine you have this network here, and the orange arrows indicate the transition probabilities between nodes, right? So if you are in node 1 and you're doing a random walk, you would transition to node 2 proportionally to the sort of the size of this arrow, and to node 3 proportionally to the size of this arrow, right? And so forth for the whole network. So if you did a random walk in this network, you could do something like ping, 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 ping around here for a second, finally go here, and finally reach the target, right? So we went from 1 to t. But we can introduce this parameter that actually provides information about the global topology to the random walk. So this is parameter lambda, and we tune it up a little bit. And now suddenly, if we want to go from 1 to t, we can increase the size of the arrows that lie along the shortest paths from 1 to t. So now if we simulate a random walk with these transition probabilities, we will be more likely to efficiently go from I to t. Just by probabilistically, we will be more likely to follow the shortest paths. And if we tune this parameter to its maximum value, we essentially only prioritize connections that lie to the shortest path from a certain target. So if you start a random walk from anywhere in this network with these transition probabilities, you would essentially flow to t via the shortest path. So this is what this idea of biasing random walks means. And in terms of biology, we could think about gradients of influence between nodes. We could think about sort of developmental axes that could be candidates that could potentially bias random walks in the brain. So the cool thing about this model is that it encapsulates in a single sort of framework the entire spectrum from routing to diffusion. And because of that, the sort of qualitative evaluation of delay, information, and energy totally depends on how you set this parameter, right? The model can be very inefficient or it can be very efficient. But what is really cool is that there seems to be a sort of nonlinearity to how this parameter affects the dynamics of these random walks. So here, this is work by Avena Koringersberger. We have, so in the red curve, we have the transmission cost, and here we have the information cost, and here we have the value of this parameter lambda. And the idea here is that as we tune this parameter and we transition from a shortest path regime, we transition from a random walk regime to a shortest path regime, the cost of transmission drops a lot faster than the cost of information transfer. So there could be, so what this is saying is that with a small bias available to guide signaling in the network, we could actually reduce a lot of the communication costs. And I think this is kind of an exciting theoretical idea. Okay, so far it's been quite sort of abstract, quite sort of a modeling flavor. So now I'm going to shift gears to go into applications. So I've introduced some of the models, but how can we use them? What are these models useful for, right? So here we have a structural connectivity matrix. It's our human connectome. Again, nodes are gray matter regions, connections are white matter connections. And what we can do is we can model communication between any pair of nodes, right? So from I to J, we select a model. As I said, this is a propagation algorithm, and we select a measure, and this helps us quantify properties of this algorithm. And what we get is this communication matrix. And what is really cool is we went from structural connectivity, which is typically sparse, and only really encodes information about nodes that are anatomically connected, right? If you have two nodes that don't have a direct white matter tract between them, this matrix doesn't really contain information about them. But once we derive a communication matrix, because we can simulate communication between nodes that are anatomically unconnected, this matrix is dense, right? It's fully instantiated. And this allows us to investigate some interesting properties about brain function. So for example, if you average across all the rows and columns of this communication matrix, you get a single value that tells you how efficiently this matrix supports communication under a putative model. If you do it at the systems level, let's say your 7 YAO networks, you can get a systems communication perspective. If you get a single row or you average it across rows, we can see how a region, the capacity of a region to send communication to send signals to the rest of the brain. Or if you average across columns, is the capacity of a region to receive signals from the rest of the brain. And using these different constructs, people have investigated relationships between these communication matrices and empirically derived data on brain function. And this is what I'm going to show you. So what are some of the applications that people have considered? The first one really was this idea of structure-function coupling, right? If we transform our structural connectivity matrix into a communication matrix that is now very dense, this communication matrix has a certain functional flavor, but does it actually correlate to neuroactivity patterns derived from empirical recordings? So this could be fMRI, EEG, MEG, SEEG, whatever you want. So the first study that kind of did this used this measure called search information, which I'm going to get to in a second. I'm going to explain this later with some examples. But it's a measure based on this idea of random walks, right? And what they did is they correlated the search information computed exclusively on the structural connectivity derived from tractography with a measure of functional connectivity from both fMRI signals. And they saw a really strong correlation between these two things. So the idea here is that regions that, based on the structure, have stronger communication channels are hypothesized to then go on and have stronger functional connectivity as well. So in other words, functional connectivity, according to this theory, is a result of communication on top of structural connectivity. And this kind of result was really the first one that people stopped and thought, okay, maybe these really simple communication models actually have a lot of explanatory power over brain function. Another thing that we can do is we can compute communication matrices at the level of individuals in a population, right? And we can try to then correlate these metrics of network communication with phenotypes. So people are doing this, for example, in the field of intelligence, right? Are people with higher measures of crystallized or fluid intelligence, do they show stronger or more efficient network communication? And you can do this between groups as well in clinical settings. I'm not going to get into the details of this direction of research, but if you're interested, go to the review and there's lots about this there. Instead, I'm going to show you something else that I find exciting. And it's this idea of mapping the effects of brain lesions, right? So if you have a focal brain lesion, you could imagine that this is going to disrupt communication between regions. And most importantly, it's not only going to disrupt communication involving the region that was lesioned, but it's going to have a global effect because perhaps that region mediated traffic between other regions in the network. So we can have a measure of disrupted global communication. And if we have a measure of disrupted functional connectivity following the lesion, we can see if these two things align. And there was a really cool paper that did this in monkeys. They use a pharmacogenetic intervention where essentially they can turn a region, a very specific region, offline. In this case, they did it with the amygdala. So what they did is they inject the region with this thing, and essentially that region is inhibited. So it loses functional connectivity to the rest of the network. And they scanned these monkeys before and after they did this. So they had a measure of the disruption in functional connectivity. And they also had a structural connectome for the macaque. So here the setup is from the structural connectome, they computed the measure of communicability, which I presented. And then they did a virtual lesion where they remove the amygdala from the structural connectome, and then they recompute communicability. So now this is a in silico prediction of how communication would be disrupted if we turn the amygdala offline. And this is really cool because first they looked at communications between the amygdala and the rest of the cortex. So here in the horizontal axis, we have the predictions of structurally based disrupted communication. And here we have the empirical changes in FC. So we can predict that really well. But not only that, they also looked at changes in functional connectivity between cortical regions that were not connected to the amygdala. So this is the idea that if you take out a region of your network, this is not only going to affect locally the network, but it's going to cause a sort of a big global network reconfiguration, which could also be predicted again by these very simple models of structurally constrained communication. So I think this is a really cool example of the utility of these models. And then you can think ahead of like, can we model the, functional deficits or network reorganization or even neuroplasticity following stroke or following TBI? Things along those lines that people are working on. Another example that I really like is modeling the propagation of brain stimulation. So you could kind of think of, let's say, transcranial magnetic stimulation, right? So TMS, which is a non-invasive technique. And a lot of what TMS wants to do is it wants to modulate the activity not only of where you put the coil and where you deliver the stimuli, but you also want to modulate the activity of deep structures, say subcortical structures in the brain. But it's unclear now how these signals are propagated through the network to lead to these deep modulations. So you could think that we have a targeted region that is not accessible non-invasively. And the problem now is, where do I stimulate so that this signal will be carried through the network to get to this region? So the communication matrices give you exactly that, right? So if you look at the columns of the communication matrix, you could say how the stimuli to the rest of the network would affect this targeted site. And if you look at the rows, it's how outgoing communication from where you stimulate will be propagated to the rest of the network. And I'm going to show you an empirical example that actually seems to work. So this is work that I did earlier this year. And we're not doing TMS, but we are modeling the propagation of direct electrical stimulation to the human brain. So we are using intracranial EEG electrodes in an epilepsy patient. So the setup is that these are patients with drug-resistant epilepsy, and they are being prepared for resective surgery, right? So this is surgery where they remove a part of their brains as a last resort that might be related to the origin of the seizures. And as part of the preparation for the surgery, they are implanted with intracranial EEG electrodes, so SEEG. And the clinicians stimulate. So these electrodes are kind of like this, and they have multiple electrode contacts along their length, along this needle, and they can both stimulate, so deliver an electrical pulse, and they can record activity, electrical activity, a millisecond resolution. So the F-Tract project, what they did, which is really cool, is they collected a lot of this data that is primarily a clinical data to inform a clinical decision, but they collected it amongst more than 550 subjects with epilepsy. And this is across Europe, across Asia, across North America. It's really, really a big data set. And it contained more than 2 million of such recordings of stimulations. And what they did is they created a very simple measure. So the schematic is the following. Imagine you stimulate this region here, this electrode contact, and you're recording at contacts 1 and 2, right? So after the stimulus here, this is the cortical evoked potential in region 1. And we see that it goes above a certain threshold, right? This threshold, we can define it kind of arbitrarily, but we can also move it around to see if the results are robust. So if it goes above the threshold, we say that this is a significant response. So we had a significant cortical evoked potential spike following the stimulation at an upstream region. So region 1 showed a significant response, but region 2, the cortical evoked potential didn't go above a certain threshold. So this was not a significant response, right? So this is an example of one stimulation. And what they did is they got all of these 550 patients, 29,000 stimulations, millions of recordings, and they condensed all of this information into a matrix that tells me across all of these people, if I stimulate region A, what was the probability of observing a significant response in region B, right? So this is the response probability matrix. Another thing that they did is to understand the median strength of this response. So when we do see a significant response, what was the size of the cortical evoked potential? So there's a red line here. And because they collated data across so many subjects, they could derive this map that had a really good cortical coverage of sort of empirical direct sort of signal propagation between regions. So this is a distribution of the stimulation sites. There's a distribution of the recording sites. It's not homogeneous because the positioning of the electrodes is informed by clinical practice, and it tends to follow regions that are involved in epilepsy, of course. But still, you get really good coverage because you're condensing the data from so many patients. So what we try to do is, yes, there's a question, Kason?
Yeah, there is one in the chat, maybe.
Oh, sorry, I'm not looking at the chat. Can you read it out for me?
Yeah, it says, are the patients awake during the stimulation?
Yes.
Okay.
Yeah, so bedside stimulation. So the goal, I'm not a clinician, but I think the main goal was to elicit a seizure or to elicit seizure-like behavior in these regions so that the doctors could get a better idea of which regions are involved with the generation of the seizures. So the patients need to be awake for that, is my understanding. By the way, this data is publicly available. It's free for everyone to download. You can just go and download it, which is very cool, I think. F-Tract project is the name, like this, F-Tract project. Thanks for the question. Again, any questions, please interrupt. So what we set out to do, now that we have this empirical measure of stimulus propagation, direct stimulation, measure at millisecond resolutions, we were like, okay, can we predict this using our very simple measures of network communication, right? And most interestingly, we thought, can we predict stimulus propagation between regions that are not anatomically connected, right? So if you stimulate here in the stimulation site, and at the recording site, you observe a response, but there's no white matter tract between these regions, this has to be a network communication problem, right? It's mediated through the underlying white matter architecture. How are these signals being propagated? So we got 5 of the sort of perhaps most popular measures that have been proposed in the literature. And they range from ideas of routing to ideas of diffusion. And we thought, which of these measures explain stimulus propagation, if any, right? If any. And here I'm just going to show you a snippet of these results. So here in this axis, we have the correlation to stimulus response. And in this axis, we have a lot of different communication measures, right? So this is SI, it's the search information, which I'm going to explain now. And CMY is communicability, which is this one. And SPE is shortest path efficiency. So this idea that things flow via the shortest path, right? And we can see here that we have a really good correlation between search information and the amplitude of stimulus response. So again, this is direct electrical stimulation measured at a millisecond resolution between regions that don't have a white matter tract between them. And we can explain the strength of this response using these very simple conceptualizations of network communication. And now I'm going to get into the detail of how this can kind of explain what's going on in the brain. Oh, no, sorry. There's still another result that I wanted to talk about, which is if you look at connected region pairs, so these are regions that are structurally connected via direct track, and we compare the performance of the communication measures with the performance of just considering the strength of the direct white matter tract between these 2 regions, we can still do better by considering communication dynamics in the network, which is to say, in my opinion, that if you stimulate a region and you measure the response in a downstream connected region, it's not only that direct connection that is carrying the stimulus, but the network topology around this direct connection also helps you predict what the stimulus is going to do downstream. And I'm going to try to explain to you quickly the interpretation of this, right? So I'm going to focus, here we used 5 measures, but I'm just going to focus on 3, which I think are the most illuminating. And this will be the last sort of empirical result that I'll show. So here we have this idea of shortest path efficiency, right? So this is kind of the, let's say, the mainstay model, what most people use. So imagine we have 3 targets, A, B, and C, and we want to measure the response at a deep region. We have 3 stimulation regions, A, B, and C, and we have a target that is the recording region where we're seeing the effect of the stimulus. And this is kind of the topology of the underlying white matter connectivity. So if the stimulus is being propagated via the shortest path between these regions, A has a very shortest path, right? Is only one intermediate connection away, is directly connected to the target. B has to traverse 2 connections, right? So it's a shortest path of length 2. And C is the furthest away. It has to traverse 3 connections, so it's the shortest path of length 3. If shortest path is a good model for stimulus propagation in the brain, we would expect the response in T after stimulating A, so RAT, to be the strongest, and then the response after stimulating B to be the second strongest, and the response after stimulating C to be the third strongest. And this is because the shortest path from A to T is the most efficient, the B to T is the second most efficient, and from C to T is the third most efficient. So this is what shortest path would predict us, right? A stronger response in A and a weaker response in C. Now let's think of this idea of search information. So technically, for the people more interested in sort of the maths, search information is the probability that a random walker will travel between two nodes via the shortest path between them. So this will be a very low probability typically, but this is how you compute it. So serendipitously travel, right, via the shortest paths. More in intuitive terms, you can think of search information as a measure of signal diffusion. And it has to do with the connectivity of nodes along the shortest paths. So let me explain. Here, this is a different example. Let's say we stimulate A. The path from A to T is filled with nodes with very high degree. So they have a lot of connections to the rest of the network, right? So A has like 5 different connections. This node here has 4 different connections to the rest of the network. B has fewer connections to the rest of the network. And this node here has also fewer connections to the rest of the network, right? So this is the path from B to T. Whereas C has very, very little connections, just one more to the rest of the network. And this node, the next one along the path, also has very few connections. So what search information is measuring is not only the length of the path, but also the propensity for the signal to diffuse to other parts of the network. And the higher the connectivity profile along the path, the stronger the tendency for signal dispersion. So here, even though A, B, and C have the same shortest path length to T, right? They're all just one intermediate region away. Search information would predict that actually C would lead to the strongest response in the targets because the connectivity along the path is so low that there's not a lot of opportunity for stimulus dispersion. B would be the intermediate, and A, because there's so many connections to the rest of the network, would lead to the weakest response in the target. So this is what search information is measuring. So not only just the shortest path, but also the local topology around this path is important. And the last case is communicability, which we already saw, which tells us that if communicability is correct, the entire network, because we are using all of the available paths and all of the available walks, should matter. So in this example, A and B are both separated from the target T via just a shortest path of 2, right? Just one intermediate connection. Sorry, one intermediate region along the quickest path. But from A to T, there's many, many efficient paths. So you have 3 choices that get you to the target very quickly. Whereas from B to T, the paths are quite long or longer. So the topology of the network as a whole makes it less efficient to diffuse from B to T than from A to T. So according to communicability, would predict a stronger response after stimulating A than after stimulating B, even though the underlying shortest path is the same. And if we go back to the results, what we found is that search information and communicability, both in the case of unconnected region pairs and anatomically connected region pairs, provided the best explanations of stimulus propagation. So this is to say that it's not only these most efficient paths that are facilitating the stimulus being carried through white matter, but the network topology around these paths really matter. And I think this is a cool result because it's saying that this diffusive flavor of communication might be more informative than this selectively accessed path flavor of communication. Okay, so just a few closing remarks to try to wrap it up. So I've talked about these network communication models. As you've seen, they're very, very simple. They're almost like toy examples of how things move in a network. But what they can afford from their simplicity is that they're very flexible. We can model brain disconnectivity. We can model structure-function coupling at rest. We can model stimulus propagation. So it's a very flexible, tractable, and interpretable framework to study brain function. There's a lot of models. I only presented you 3 or 4, and we're still not sure which of these models and measures are useful. And I think the tendency of the field is to always propose new measures instead of focusing on empirically validating the ones we already have. So this, I think, is an important kind of gap in the research direction that has been going on, especially in network neuroscience. So I think efforts to validate models is important. I think in that direction, interventional and longitudinal data offer a really cool opportunity. So by interventional, I mean where you go and you disconnect a brain region or you stimulate a brain region, you have a direct manipulation of the system. Longitudinal could be data where you follow people after a stroke to understand network reorganization from the perspective of communication, for instance, or you try to model the spread of neurodegeneration in Alzheimer's, for example, which is something I didn't even talk about, but it's another application of communication models. So I think this flavor of modeling goes away mere descriptions of brain networks, which have been very useful and have been the focus so far. But now I think it's time to use these models for prediction and prognosis. And of course, as I said, these models are very simple. And because they are so flexible, they abstract away neurobiological detail. And one of the downsides of that is that they don't really tell us about specific mechanisms of brain function, right? They're more phenomenological in their nature, they're mechanistic. And I think a really cool direction to try to expand on that is to integrate different modeling domains, right? There's really cool work that tries to integrate graph theory with dynamic models, with control models, with information theory models. And I think this is a really cool direction. And that's all for me. Again, this is all packed into this review. If you're interested, read the review, send me questions on my email. And that's it.
Okay.